What is epistatic selection and what is recombinant inbred line? As Ernst said, this is a joint work with Ernst. He's my supervisor. What is recombinant inbred line? Recombinant inbred line is a study design that can be for animals or plants. So the idea is that typically is that recombinant inbred line derived from two inbred parents. So by inbred, I mean parent, for example, the mother, all the genome of the, for example, parent 1 or the mother is red, all 0, coded as 0, and the father is all 1, coded green, coded as 1. So they cross these two plants, or it could be mouse. So they cross, and then they will have F1 generation. In the F1 generation, the half of the genome of the F1 will be from parent 1, and the other half will be parent 2. And then in case of the plant, they do or animal, they will keep crossing with a sibling or selfing, and then they keep doing it, and then at a certain generation they stop, and then they will have different types of plants. For example, if we look at the genome of this plant, we will see that the first section, this, for example, the location 1 till, for example, location 10 comes from the mother, and the location 11 till location 40 comes from the father, and etc. And there will be also heterozygous location, which half mother and half father. So you see that, for example, here there is this kind of represented genetic linkage. Because when, in the meiosis, when they cross the plant, the parents, they had the markers have a tendency to transmit it together from the one of the parents to the offspring. That's the reason we see a series of 1 and a series of 0. So this is just a cartoon example. Just assume that each node here is a marker in the chromosome 1 of the offspring of the child, and then each edge here which says that marker 1 is correlated with marker 2, should be associated with marker 2 in the genome of the offspring. This is exactly this links here shows the genetic linkage. This is because of the series of 1 that we see in the genome of the offspring, and then 0, 0, 0, and then 1, 1, 1. So this calls genetic linkage, these links here. Also the same goes to the chromosome 2 and etc., all the chromosome. But we have a law in the genetic, it's called the inheritance law or independent assortment law, which says that markers markers in different chromosomes should segregate independently. But sometimes when they do this crossing, they will see that, for example, some of the offspring, they die, or they have some kind of disorders. So, and then the plant or the child will not be survive. So this called epistatic selection, which shows as the correlation between the markers in different chromosomes. So if epistatics happens, then it will show as a correlation between markers in different chromosomes, and then for example here marker 2 and marker 15 in different chromosomes, they affect the viability of the offspring. So here, for example, you see here, this is in the lab, they did it in the lab. So some plants, for example here, this plant here, the combination of the genotype caused that he has a long root. This one doing fine, this has a weak root, or it shows here the embryolocality. So certain combination of the genomes don't function well together when brought to the genome of the offspring. So our aim is to detect the high-dimensional epistatic selection in the genome of recombinant inbred line, and our approach, we are going to extend the graphical models for the discrete ordinal variables, and we are interested to check to see that this epistatic selection, this association based on conditional independency, and among the genetic markers. And the nice thing is that there are no methods so far about to check this epistatic selection for polyploid species. Polyploid means diploids, tetraploids, or when we have 2 copies of chromosomes, 3 copies, or 4 copies of chromosomes, something like some plants, some potato, tomato, or also some diploid species. So there are people, they usually do in diploid species is like they do hypothesis testing, just to test 2 markers 2 by 2 and then do Bonferroni correction, and then they end up with no significance. And also in a high dimensional area, the hypothesis test doesn't work well. So, but what do we mean by conditional independence? Probably you know all, but just pointing to it. So, for example, here, variable 2 and variable 4, we study the correlation between them given the rest of the variables. So, if there is no link here, it shows that variable 2 and variable 4 are conditionally independent given the rest of the variables. And for example, 2 and 4 here, the same goes to for variable 2 and 3. So they are conditionally independent given the rest of the variables. The nice things about the graphical model is that if the variables come from the normal distribution, then we have a nice property that precision matrix. So this is the variables, and this is a precision matrix here. And the precision matrix, when we estimate the precision matrix contains all the information of the conditional independencies. So we are interested to find the theta here in our model. Just to point to what kind of data do we have. For example, in terms of for the diploid species, we have 3 levels. So for example, 0 when the genotype of the offspring comes from the parent, for example, from the mother, and the other one is genotype of the offspring comes from the father, and the one here is in between, half from mother and half from the father. The challenges here is that we want to recover graphical model for discrete ordinal data in high dimensional settings, and we have a large p small n, and also the traditional work that multiple testing that I pointed to, so it's under power. Now I'm going to talk about our model. So we have a Gaussian Copula model, and we use it in our model. So assume that we have a latent variable which has a mean 0 and the precision matrix theta, and the diagonal of our covariance matrix is all 1. So we assume that there is this underlying latent variable, and so we have these cutoffs which is not known. We can calculate these cutoffs, and then we can rewrite the data in this. We can rewrite the data in this way. That is the relationship between the data and the latent variable. And then we transfer the data. This is our data, and this is the latent. We transfer the data based on these marginals. So we have marginals, and we have this discrete, and then we have marginals, and we transfer it to the continuous space. And so if we want to infer the likelihood of the observations. So this is the likelihood. It's involved in here the linear integration. Also, it doesn't have a closed form. So we address this issue by using penalized EM algorithm. In the penalized EM algorithm, we use Q function. Penalized Q function is the expectation of the joint likelihood given the data and update version of the precision matrix. And the M step is the maximization step, which we update the theta in each step. And we are interested we are working high dimension. We are interested in the exact shrinkage. So we want to use L1 norm. We want to exact 0, so we use L1 norm. But the thing here is that the Q function here, it looks like this, and we still need to calculate this part. We propose to go in 2 ways. I'm not going to talk about the formula at all, just going to the detail. But for the Gibbs sampling, the idea is that it's simple. So we generate the random. We sample the from random truncated multivariate distribution, and then we calculate the expectation of from our Gibbs sampling. And there is another idea. This approximation estimation. This is the approximation. This term here gives the off diagonals of this covariance matrix, and also this term here gives us the diagonal. So these 2 terms here, this just gives a number for the off diagonal, and this also gives a number for the diagonal. And then in this way, we can approximate the precision, the covariance matrix in the ES step of the EM algorithm. So we do this EM, and then for its certain number of iterations, we have a criteria to stop our EM. So for the checking a convergence, and at the convergence point, So we want to do the model selection. So we work with a joint likelihood. Now we are interested to find the likelihood of the observations. So we have this this term here gives us the likelihood, bring back us the likelihood of the observations. So we already have the Q function. Actually, we have a penalized Q function, but we are here, we use a Q function. We took the penalty out, and then also we can calculate at the EM convergence the H function. And the H function is simply the expectation of the conditional likelihood, log likelihood. So because we are interested to find the graph structure, we prefer to use the EBIC, BIC rather than AIC or other information criteria. So this part is a likelihood of observation, and this is a bias term, and for the EBIC follows this one. And for the penalty term, that we have a minimum EBIC, that's a kind of a winner model, or we have it's our best model. So this is a general view of our model, and now we did some simulations, and we also compared it with other methods. So the blue one and the red one is our method, and the two green bar is two other methods. We compared with P 150 and N is 50, and the number of levels is 5. So, and the two other methods is based on Ken delta and non-paranormal score, normal score. So with the area under the curve, ROC curve, shows us the performance of the method. So our method outperforms the other methods based on the ROC curve. And also the same scenario when we take the number of the levels higher, so our method performs again better. So now we are going to implement this method to the real data. The first real data is Arabidopsis. So this plant is by crossing Colombia. It's one of this plant in Brazil, I think. I don't remember exactly, but the other one is CVI. So these two are parents of the two parents, and then the offspring has a 90 genotype, 90 markers, and for 367 plants or individuals, and the data is 0, 1, 2, 0, 1 is from the genotype offspring comes from Colombia, and the other the 2 is genotype of the offspring comes from CVI, and the 1 is half-half. Of course, we have also missing genotype. Is very routine in genetics. So then this plant, we know that has 5 chromosomes. So this is the output. So each color here shows 1 chromosome. So we have in total, we gain from our method all 4 or 5 chromosomes. And when in the second slide, I talk about this genetic linkage. So we see this genetic linkage here. They are highly conditionally correlated within the chromosomes. So this is also the just image of this network. So we see here really 5. You can even exactly distinguish these 5 chromosomes. So this is nice. So to have these links here, but the things that we are interested to epistatic study, people can go study here about what's going on here if there is a cis interaction. But we are interested to the off diagonal because epistasis happens there. So we are interested to find the genomic location in the offspring that cause these disorders in the plant or in animal. So this data, why we chose this data, because this data is really well studied in the lab, and people in David Bicart, so also they published it in a science paper, is very well studied. So they find that in the lab that this genomic location that we found here cause weak root in the plant. Also, the other one that we found it caused embryonal lethality in the plant. So because they did it in the lab, also they did hypothesis testing, so they couldn't find other few more links that we found it. So this could be other disorders in the plant that they couldn't find it, and we detected with our method. So this is like just to verify our method, and we implemented for the MACE data. So where P is the number of the markers around 1,106, and the number of the individuals is 193. So the maize has 10 chromosomes, and they expect much more epistasis happens in this species because it's outcrossing. so they cannot really control it. So that's the reason we see, so this chromosome, we see more between chromosomes, the links here. So it's an idea that it's our idea to send this our findings to the lab in Cornell University, that they have a really big grant to study the maize genotype, the maize epistasis in the maize. So also this very interesting was, while I was talking with one of them in the conference, it was very interesting for them why this chromosome here is completely separated and a lot of stuff going on here. So this transchromosomal relationship between different chromosomes, the existence of such transchromosomal relationship reveal aberrant marker-marker association that are due to the epistatic selection, and then we see some kind of disorders in the plant. So this was the whole structure, and so to wrap up is our aim was to detect high-dimensional epistatic selection and in the genome of recombinant inbred line for any polyploid species, could be tetraploid or any kind of species. For this aim, we extend graphical models for the discrete order of variables, and then we are interested to find this epistasis based on conditional independency. We verified our model in Arabidopsis, and we employed our method to find the genomic region in the maize, which may lead to the maize disorder. So we have, soon our package will be online. It's a multi-core package, so it's called multi-epi loci, and soon it will be online in the CRAN. Yes, thank you.
Okay, questions for Pariya? Yep.
I mean, because of the linkage disequilibrium, you always have a lot of correlation between nearby sites. Did you do anything to sort of, I mean, is this something, because you showed the matrix where this was indeed the case, is this something that just emerges from your data analysis, or did you have to somehow account for that prior knowledge in the model?
So if I understood correctly, you mean you are talking about, for example, the findings that we have here, how we are certain about this?
No, actually, what I'm saying is you will know, like, even it makes perfect sense that you will see a lot of correlation on the main diagonal.
Yes, that's for sure.
Because, and so the question is, does your model somehow bias is your model somehow biased towards finding those solutions, or in order to increase power, or is this something that just naturally emerges without any sort of constraints?
No, I don't have any constraints. So this is a natural. So it's just we implemented. I don't have any threshold. This is just finding. We put the data in the package, and then there is no threshold. Just it shows that they are conditionally independent. These two loci given the rest of the markers in the genome. Yeah.
Maybe just a follow-up question. You've also done some work on potatoes where you actually do not know the locations of the markers necessarily.
Oh yeah, that's true. So there's also another application that we don't know the location. Just they give us a bunch of markers, and then they tell us, okay, construct a genetic map. So tell us which marker is where, and just tell us, and also how many, even they don't know how many chromosomes do they have. It's not in potato, but other applications. So in this way, we can implement this method, and then on top of this method, we do some extension. We detect communities, and also we do some ordering markers to kind of to construct a genetic map of the species. So that would be the second application of our method. Yeah.
Other questions? Okay, if there's no further questions, then let's thank Pariya again. And as I said, T will be